Good morning, students. I'm here with the pharmacodynamics and its concepts, part 2. Already you have understood the background behind the pharmacodynamics in the part 1. So now we will discuss some more important issues. This is the same diagram what I have shown you in the part 1, which gives you the clear idea that how the pharmacodynamics you know enters into the pharmacodynamical process to get the final effect of the drug. So we'll move on with the receptor concept. When we say the receptor concept, it starts way back in 80s when the word receptor has been coined, and from that time itself the various scientists were speculating about the drug's effect, right? See, that drug goes into the body and it may bind with this, or it may do this way or that way, like some speculations were there. And in that way, slowly various discussions happened, various studies happened, and the a concept emerged. So that concept I'm bringing to you, that's as a receptor concept. So whenever you have the why you need to understand the receptor concept, because you need to understand the drug effect. Once you are interested to know what the drug effect and how that effect comes, so you have to know about the receptor concept. So what do you understand by the drug effect here? So there's a one definition of drug effect, like it is the ultimate change in biological function brought about as a consequence of drug action. So already in the part 1 you have seen the drug action. So by that drug action, whatever the final change or the final biological change, whatever happened because of the drug, called as the drug effect, right? And this happens through a series of intermediate steps. It's not like a one drug and there is a one effect. There's a chain of events happening. So by that you will get a drug effect. So to know that drug effect, you have to understand the concept of receptors first. Uh, Scientist Dr. Rang have very beautifully narrated, or you can say poetically thought about the receptors, and he expressed it's around in 1964. That time he has because a lot of talk of receptors were going on, right? So there were no concrete proofs were there. So he has narrated it in such a way: a beautiful but remote lady. Means it's a like he has written a letter to the receptor and has said the receptor as a beautiful but remote lady. To most of the modern pharmacologists, the receptor is like a beautiful but remote lady. He has written her many a letter, and quite often she has answered the letters. From these answers, the pharmacologist have built himself an image of this fair lady. He cannot, however, truly claim ever to have seen her, although one day he may do so. So this is a kind of uh, poetic description they have given, that they are trying to reach to the answers for the receptors, but yet receptor is not reachable for them. And what one day they are hopeful that one day they'll get it. This is a that time. Now we are in the good era that we know everything about the receptors, right? So this is written by the D.K.D. Jong actually. For him and others, the cell receptor was something of an enigma on that time. They know that there is something like this, but they were not able to find it. So it's a kind of enigma for them. There was one scientist known as John Newport Langley. So he, from the Cambridge, he was, he has given that yes, there is a drug, definitely it acts on the specific site. So that from there the idea has emerged. That time itself, Paul Ehrlich was there. He was the greatest scientist. He has coined the term receptor, right? And later on, the Alfred Joseph Clarke has he was working from in the King's College London. So he has given the so many other informations which have helped to build the receptor concept later. So this is a little brief history what was going on that time. Now I tell you another very important point that's called chemical signaling. Signaling word means there are some signals originated from somewhere, received by something, and enacted upon that. Okay, something has been emerged which has been received as a signal, and that signal, once been received, been enacted in a certain way, right? So that is the signaling process. And this, without understanding the signaling mechanism, we cannot understand the receptor concept. Actually, the signaling mechanism is the broader term of the receptor concept, if you see, right? Because it is inherent into the receptor process. So signaling is very, very important. This signaling word also sometime what called referred as a communication. So there we can say as a communication process, because one thing, one protein, one chemical, one factor is communicating with the other factor, and the other factor will communicate to other factor. Like that, this communication goes on. So same thing, this communication we call it as signaling as well. Here we are talking about the signaling by the chemicals. So that's why the chemicals which are involved in between, and they are passing on the signals from here and there. So they are playing a very important role in the whole communication process, that we call it as a chemical signaling. So as I told you, is the main mechanism which biological function is controlled at all levels, from the single cell to the whole organism. So by this, the whole biological process has been controlled. Next is the chemical recognition is the function of receptor. Means what? The receptors will identify that particular chemical, and then they will react, right? That chemical can be your drug, that chemical can be hormone, that chemical can be any other factor in the body, any other chemical in the body, neurotransmitters in the body, right? So these all are the chemicals what the receptor identifies, right? So that's why receptor is also the part of that communication process, because receptor is the one who has first received some signal as a chemical, and then that has been transduced in various other proteins, right? So which, in addition to recognizing indigenous chemical signals that I told you, are also the target of many important experimental and therapeutic targets. Receptors therefore lie at the heart of pharmacology. So from beginning I'm telling to you that receptor concept, without a receptor concept knowledge, there is no good pharmacologist will be. So if you want to be a good pharmacologist, you have to know the receptor concept well. That's why the receptor lies at the heart of the pharmacology. That is written by H.P. Rank. Now we'll talk about the drug receptor interaction. Now we'll reach to the first point of this signaling mechanism or this receptor concept, where a receptor is going to interact with the drug, or the drug is going to interact with the receptor. When you have taken the drug goes inside and go into near to the cell, because it has to enter into the cell and then do something. So in that case, it interacts first with the receptor, because as I told you the definition of receptor, receptors who receives. So receptors are there, and they are nothing but the protein structures that I will tell you later in the presentation, where we'll see the different receptors. So receptors are the different protein structures. They will receive the drug, or they are the first who will bind with the drug, right? So let's see what the drug receptor interaction brings out. Here one formula is given like drug receptor interaction. You can say D as a drug and R as a receptor. So when drug and receptor get combines, it gives you DR, means is a drug receptor complex, right? It's called drug receptor complex. And then what you will get at the end, when there is a drug receptor complex, you will get drug response, okay? Or you can say the response of the drug. Binding of a drug to the receptor is usually reversible. Another point is very important, that this binding D plus R, this binding is usually reversible, but in some cases it is irreversible as well. A receptor is analogous to switch in that it has two configuration, one on and off. So you keep in mind, receptors are the one who keeps this whole process of communication switch on or switch off, right? When they are activated, the whole process get activated. If they are not activated, the whole process is switched off. So that's what the this receptor and drug complex, or this thing does, right? Now we'll talk about the factors governing drug action. This is very important to understand what are those factors which are actually changing the drug action. These are two important components I have already discussed before. Just here I have given again to link it with affinity and intrinsic activity. So affinity is a measure of the tightness that a drug binds to the receptor. So again, affinity is just a binding of a drug with the receptor, right? How it binds, right? A high affinity means that a low concentration of drug is needed. Understand this point very well. High affinity means that a low concentration of drug is needed to occupy receptor sites, whereas a low affinity means that much higher concentration of drug is needed. Understand this point very well. When we say the drug is having high affinity, it means even in low concentration, right, it binds with the receptor well. While we say low affinity means we need more concentration of a drug to get it bind properly. Okay. Intrinsic activity is a measure of the ability of a drug. Again, concentrate here because a very important definition. Intrinsic activity is a measure of the ability of a drug once bound to the receptor. Means once the drug binds with the receptor, now the ability of a drug to produce the response is you are calculating. So if drug is producing the response, you can say yes, there is a intrinsic activity, right? If not producing, means no intrinsic activity. Affinity and intrinsic activity are independent properties of drugs, right? They are not dependent on each other. Maybe drug have affinity but no intrinsic activity, as it in case of the antagonist. Maybe drug have both in case of the agonist, right? So like that, but definitely there is no such case where there is no binding, no affinity but intrinsic activity. That case not happens, because first you need to get bind, the drug need to get bind with the receptor. Agonists have both affinity, the ability to bind to the receptor, as well as intrinsic activity, the ability to produce a measurable effect. Antagonists, on the other hand, only have affinity for the receptor. Affinity allows the agonist to bind the receptor. Intrinsic activity allows the bound agonist to activate or turn its receptor function. In this way also you can understand the affinity and the intrinsic activity. Okay. I have given these things so many times, so because I want you to understand and remember what the intrinsic activity and what the affinity means. When the drug is binding, you can see the drug can bind by using the following forces: covalent bonds, ionic bonds, hydrogen bonds, hydrophobic bonds, or the van der Waals bonds, right? You can say covalent bonds are there. So like that, its binding needs different forces. Now to understand the affinity, here some pictorials are there which helps you out to understand. You can see in the top portion the D is a drug, R is a receptor, D and R gets uh, in touch with each other and gives the DR as a drug receptor concept. In this you have two rate constants K1 and K minus 1, because K minus 1 shows the reverse direction, right, means DR get dissociated again. So K1 is shows you the association where D and R gets bind and makes the DR, and the K minus 1 shows the dissociation where D and R get separated again. Okay. If you need to know the affinity, then you can say K1 by K minus 1, and Kd is equal to K minus 1 by K1. So like that, these pictorials will give you the information. You can see on the right side here you can find the drug association has been shown, that is K1, and here drug dissociation has been shown, that is K minus 1. This is a reversible reaction, and when at equilibrium the rate of drug receptor complex formation is equal to the rate of drug receptor complex dissociation, right. So here it shows the equilibrium condition. The rate of formation of the drug receptor complex is described by K1, and the rate to which drug and receptor complex get dissociates showing as K minus 1. Okay. Receptors largely determine the quantitative relations between dose or concentration of drug and pharmacological effects. So again, a very important point to understand here, receptors largely determines the quantitative relations between dose or concentration. So drug, what dose it has been given, what concentration it has been given, it determines what pharmacological effect, and you can measure the quantitative relationship between these two. Okay. The molecular size, another important factor which is coming here, that is molecular size, shape and electrical charges of a drug. So when we make a drug, as a pharmacist or as a pharmaceutics person, when you design a drug or a formulation, you have to take it in mind the molecular size, the shape and electrical charges of a drug molecule, right. That's why it's very important to study the drug molecule before finalizing a formulation, and that determines whether and with what affinity it will bind to a particular receptor. So by that you are defining that how tightly it is going to bind with the receptor. It's going to be reversible, it's going to be irreversible, and whatever the way it's going to bind, that you define before itself, right. Another very important concept I want to introduce to you, that is partial and full agonist. You can think in mind that what do you mean by the partial agonist. Agonist is okay, right. One drug goes, binds and gives the effect, that drug called as agonist. But what do you mean by the partial agonist here? Agonists may differ in how tightly they bind to the receptors, right. Again, keep in mind, once you say the drugs binding, when you are talking about the drugs binding, right, it means you are talking about the potency of the drug. You've seen now in before previous slide what I said, that low affinity and high affinity defines at what concentration you need, right. So any drug which needs a low concentration to produce its effect, it means it is having good binding by that low concentration. That's why it has a high affinity. Okay. So high affinity drugs are actually having high potency. Okay. So always understand in potency word in this term. Potency always defined with the ability of binding, right. And then ability of binding defines how much concentration they use to get bind with it, right. So that's what how tightly they bind to their receptors and the effect they produce. So after binding there, what effect they are producing, that effect, it defines the efficacy of the drug. Okay. It means if you go back into it, you will understand the efficacy is related with the potency related with the binding defines the concentration, at what concentration it binds, right? So like that it goes. Okay. Some drugs may bind very tightly, called as are highly potent drugs, but produce only a modest effect, maybe the low efficacy. Okay, so maybe that's why they have said initially the binding and the intrinsic activity are two independent terms. They are not related with each other. So this is possible that the drug may be having a high potency but the modest efficacy. Low efficacy drugs are termed partial agonists. This is what you have to understand. Low efficacy drugs are termed partial agonists, while the structurally related drugs which produce a full effect are called full agonists. That's why a term, because of the partial agonist, a term came full agonist, right? So those drugs which you call it as actually as an agonist, they are actually full agonist, right? So any drug which is going to bind very well, have a good affinity, but low intrinsic activity, right? So that you call it as low efficacy. That kind of drugs are the partial agonists, right? Because I have already made you clear about the partial agonists, because they are not properly binding to the active site, right? They bind and occupy the active site, not allowing the true agonist to bind with it, but not binding fully. So they are not able to initiate the optimum effect by the drug. Here you can understand with this graph, you can see the blue color line shows the partial agonist, which has been a drug C, and red color line is the full agonist, and the green dotted line is the also full agonist. So you can see there are three drugs have been shown, but they are variable in their what called drug dose as well as the effect. You can see the one side is maximum effect and one side is drug. So you see the red one needs less drug and produces the high effect. Okay, so that's why we call it as full agonist. Second, if you see the green one needs more drug but gives you the similar effect what the A has given, right? So you can see comparatively is my question to you, which drug is having high potency, A or B? Also, which drug has the high efficacy, A or B, right? Because you just refer for this answer, you refer the previous slide, then you will understand what I am talking. Okay, and the partial agonist, if you see, it needs a dose in between the A and B, but response, if you see the drug's effect is somewhere very low. If you see the drug's effect of this one is somewhere very low, it reached here only, right? While these are having here and here. So the effect is very low, while the dose used by the C is almost in between, or you can say almost the same what the full agonist B drug has been used, right? So you could have understood that what do you mean by the partial agonist here. Now another clarification I wanted to give you here, then how can an agonist be a partial? So this is little one more justification coming here, that how agonist can be a partial. So there are two theories have been given. One is the partial agonist may fit the receptor binding site well, but is less able to promote the receptor conformational change leading to transduction. So one theory says that they may fit into the receptor site properly, but they are not able to change into the receptor molecule, right? Maybe they are not able to make some conformational change. When the word conformation comes, it means the change is that significant, the significant change which can activate or you can that can give the status of activated receptor, right? That can give the status of activation to the receptor, right? That's what called the conformational changes. So what happens, first theory, they are fitting into it, but they are not able to produce the conformation change into the receptor. That's why the responses will not be sufficient or optimum. The second theory says the receptor may isomerize between two states. You know that the receptors have the tendency of having the active state and inactive state. If the A form has high affinity for the agonist L, suppose that here they have said the A form has the high affinity to the receptor for the affinity for the agonist L, the equilibrium will be shifted in favor of AL and full activation might be achieved, right? So if it is an active form, you can see here A plus L gives you AL if it is an active form, right? While if the I and A form of the receptor share similar affinities for L, both AL and IL will be formed, but only AL will produce an effect and the agonist will appear partial. So what is happening here? IL is an inactive form of the receptor, bind with L, your agonist, right? So what you have got? L plus I, right? You have not got complex, the binding not happen, right? Or you can say, and that way you won't get the proper response. Okay, so this is way the partial agonists are actually working. Now there are certain theories which has been given for the drug receptor concept and still these theories exist, and but there are certain theories which has been widely accepted. So I will tell you, and from the beginning, as I told you, the scientists were finding out that how it happens. So here is one hypothesis given by the Clark. He was one of the known, very renowned name in the receptor or molecular studies. What he said, the pharmacologic effect of the drug depends on the percentage of the receptor occupied. What they said, suppose that there are 10 receptors, right? So 10 receptors available and drug goes and binds with 5, right? So what they are saying, the effect of the drug will be determined by the percentage of the receptors occupied. Okay, so this was the theory given by this. If receptors are occupied, maximum effect is obtained, right? Chemical binding follow the law of mass action. So their hypothesis, the Clark's hypothesis, was based on the law of mass action, and it's not completely wrong. In some way, it's right as well, but not defines the whole process of the receptor concept. This is the Clark's simple occupation theory. What Clark has said this theory? He said simple occupation theory, which was based on the law of mass action. You can see the D plus R gives dr, and Kd is the equilibrium dissociation constant, and Ka is an equilibrium association constant, right? Already I have shown you in previous slides that what do you mean by the dissociation and what do you mean by the association. So here the constants are playing the role, Kd and Ka. So if you need to know the Kd, you have to do K2 by K1, and Ka, if you want to know, then K1 by K2, you can do that, right? So D is the drug, R is a receptor, right? So Dr is a receptor complex. So this way the law of mass action has been defined by the Clark simple occupation theory. Second theory was given by the actually the same Clark itself has given the assumption of his theory, what the previous slide has shown. What it talks, what gives the assumption. Responses to drug result from formation of reversible complexes with receptors. So this is the one thing what they understood by this, that the responses to drugs result from formation of reversible complexes with receptors, right? So whenever D plus R binds, they forms a Dr and it's a reversible complex. One molecule of drug occupies one receptor. This is another concept what they derived from this, that one molecule of drug occupies a one receptor. This is still true. The response to a drug is proportional to the number of receptor occupied. Another point he said, the response to a drug is proportional to the number of receptor occupied. As many a number of receptor will occupy, the response will go up. A maximum response to a drug occurs when all receptors are occupied. This is what the Clark said, but it's not happens every time, right? That all the receptors get occupied. Maybe I'll show you further some graphs where only a fifth means even 100% receptors are not need to give you the 100% response, right? So 100% response can be achieved even before that. Okay, so that's why this point is not always true, but it was a good theory. The total amount of drug bound to receptor is negligible relative to the total amount of drug present. This is also actually correct. The drug what you have taken, it is all, maybe you have taken a 100 milligram drug. So this 100 milligram drug is not going to give you the total response. Maybe out of this 100 milligram, only 1 milligram drug gives you the response. Okay, so that depends. Later, you have already understood in pharmacokinetic that what happens to the rest of the 90% drug, right? So that you can understand by the metabolism process and the pharmacokinetical process. So this is the way what the assumption of Clark has been brought into. Arens and Stephenson have given another theory, which they also call it as occupation theory. Effectiveness of a drug lasts as long as the receptor is occupied. So this is another intervention of that thing. What they said, if the drug has bind to the receptor, as long as it is bind with that, the effectiveness or the drug's effect will continue. Okay, many substances possess different effects. Some have high affinity for a receptor, some have low affinity, and some are not effective, and those ineffective substances block or inhibit the receptor. So like that, here they have given one clue of agonist antagonist kind of concept, right? Now, Patton has given another hypothesis. What it says, the effectiveness of a drug does not depend on the actual occupation of the receptor, but by obtaining proper stimulus. Now, here the Patton is little, differentiating from the other two. What he is saying, effectiveness of a drug does not depend on the actual occupation of the receptor. He said that effectiveness is not depending on the occupation or binding of the receptor, while it depends on the proper by obtaining proper stimulus, right? So what stimulus is being received, right? In other way, later when you understand this point, somewhere will go and will be true what the previous theories have said, right? But here when they have written it, sounds little different. This is, they call it as a rate theory. This is, I think, the one very widely accepted theory, which we call it as the lock and key hypothesis, Fisher's, given by Fisher. The drug molecule must fit into the receptor like a key fits into the lock. And this is really a good concept, or very simple way of explaining the intrinsic activity of the drug, right? Means unless until the drug is not going to fit into the grooves as the key fits into the lock, lock is not going to open. Means here lock is opening means receptor get activation. Okay, and when that happens, then only the intrinsic activity has been shown. So every lock has its own key. Same way every receptor has its own agonist or its ligand. If the key is not precise, the lock does not open, right? If the correct ligand is not binding, the receptor won't get activation. The drug is the key that has to fit the target especially and productively. So this is the way they have explained the lock and key hypothesis for the receptor concept. This is also very good theory was there, Cosland's induced fit hypothesis. This is really effective theory, induced fit hypothesis. You can see in the picture here, substrate A and substrate B, which are binding on the enzyme C here, and when they bind completely, then what happens? Once they bind, they have induced the enzyme's activation, right? Like that, the enzyme get activated. After the activation, maybe it is moving off from it. So what it says, at least two steps. Example, step 1 is initial binding and step 2 is a change in structure of the receptor. So always important, I told you beginning as well, It is very important the drug has to bind with the receptor as well as give the conformational change in the structure. Receptor is flexible, can wrap around the drug. The zipper model is extreme case of induced fit. Zipper model means like a, you see the zip, and a zipper, if you see, once you close the zip, it gets fit very nicely. Nothing can come out of the packet if the zip is closed. So the receptors and the drug will get fit like this. Okay, but that is a, what they have said is an extreme case of the induced fit, but concept is similar. If you see here, actually this way a drug get fit into the groove. Okay, all intermediate cases do exist in the nature. So I hope you understood the induced fit hypothesis. Now we'll move on to the dose response relationship. This is very important because now whatever the theories you have studied, you need to test them. And for testing these theories, this dose response relationship is very important. If you understand this concept, it will be easy for you next year to do the experimentations of the drugs, right? You are going to do the bioassays. There also a dose response relationship is going to be tested every time. So in general, biological responses to drug are graded. So this, now I'm going to introduce few new terms to you. One term is called the graded. Graded means response continuously increases as the administered dose continuously increases, right? Point 1 given you one response. Suppose that you have given the point 1 drug, right? So you got a response like this. Then you given point 2, you got the response like this. You given point 4, you got the response like this. You got the point 6 response like this. So what happens, this is called the graded response, right? As you are increasing the dose, the response is increasing. Okay, when a graded dose response relationship exists, the response to the drug is directly related to the number of receptors with which the drug is effectively interacts. So in the previous see here, either you talk about here, 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 here. So what happens here, number of receptor occupied are less, here little more, here little more, here little more. Okay, so what happens, this concept comes in the graded response. Once you have given the point 1, suppose that for example there are 50 receptors available, right? When you have given drug point 1, maybe 10 receptors occupied, or maybe 15 receptor occupied, right? Then you got one response. Then you have given point 2. Now this more concentration has given you more receptor occupation, maybe now got 25. Then you got point 3, you got next 30, 35. Like that, the receptors occupation is increasing, and same way your response is also increasing. This is one of the one rule or the belief or the concept behind the pharmacology. Now for this dose response relationship, now you understood the dose response relationship. There are other relationships will come further, which is based on this dose response relationship only. You have seen the graded, you will understand the quantal responses also, it will come later. So but the concept will be same with that. So now we'll see, if you want to draw a curve for the drug receptor interaction, how it will look like. Because now in the third year pharmacology experimentations, you will see these, you will you are going to get these curves every day. So here for one example, a clinical study may examine the effect of increasing amount of analgesic or pain threshold. Here they have given an example. Suppose that you have analgesic drug and there is a pain threshold. Now you want to see, when you give a drug, at what point of time the pain will be relieved. It means the effect of the drug you want. So how much drug you have to give to get relieved of from the pain. So that's an example you have set. To present the data, the concentration of the drug would be plotted on the x-axis. So here what happens, you are going to plot a graph, right? So you will plot here x-axis the concentration, here concentration you are drawing, and pain threshold will be presented. Like here you will draw the effect of the drug, right? Here you will draw the effect of the drug. So like that you will see that how much concentration given and how much pain has been relieved. Like that you are drawing. So that will come as a graph for you or a curve for you. A plot of drug concentration D versus effect E is a rectangular hyperbola. So when you draw this, what you will get? A rectangular hyperbola, right? This is a type of curve you will get. This is because there are a finite number of receptors. So one thing is pretty sure that there are finite number of receptors available. Hence, the response must eventually reach a maximum, right? The necessitates a long x-axis. So what you need? You need a very long x-axis because finite receptors are there and it reaches to maximum dose. So that is not possible to draw. That much receptors are there. You can't go up, keep on increasing. So to overcome this problem, what happens? The log of the drug concentration has been plotted versus the effect. So they are not drawing the dose of the drug and the effect, while log dose they are drawing. So you can bring under the graph. A plot of the log of D versus E is a sigmoid curve. So you in place of rectangular hyperbola, you will get a sigmoid curve, which will be more easy to study. You can see here log dose response curve has been shown. One is the dose response curve. You can see this is the dose response curve and this is the log dose response curve, right? You see here this is an hyperbola response and here you can get a sigmoid one, right? So you can understand here the dose, as your the dose is in the particular dose, it's going like this, and here if you see the dose, if you are given that particular dose, you will get that particular response, right? Even here it's easy to draw the ED50. ED50 means effective dose 50, right? Suppose that you have 100 patients, so at which dose 50% of the patients have got their pain relieved, okay? So that dose we call it as effective dose 50, right? So ED50, always remember this. So the position and shape of the log dose response curve is dependent on the affinity of the ligand for the receptor and its intrinsic activity. Affinity determines the position of the dose response curve. See, now you have already understood the concept of the affinity and the intrinsic activity. So what affinity does? Affinity determines the position of the dose response curve on the x-axis, right? So on the x-axis where the what call the uh, position dose response curve, where it lies, right? It lies here, it lies here, here, what on the x-axis, while the intrinsic activity effect in the magnitude of the response, and here the effect will be lying, okay, this way. So this way the log dose and dose response comes. Here also one more graph. Here you can see percent of maximum effect has been shown. So EC50 and the here also the two graphs have been shown. One is the dose and another is the log dose. So in the log dose you can see, you can find see EC50 the value will come almost same in both the cases, right? But the drawing will be easy by the log dose. Now here another graph is there which can help you out to understand the drug potency and the efficacy. Very important graph to understand. Efficacy is the maximum effect of a drug. Potency is a competitive measure, refers to the different doses of the drug, two drugs needed to produce the same effect. Okay. So normally what happens, we plot that data as effect versus drug. Effect, you can see we have plotted already as effect versus drug. Okay. This makes it easier to determine EC50 values and to compare the drug efficacy and potency. So here if you are using different drugs like that, they have used it here, drug A, drug B, drug C. So three, and they are comparing the potency and efficacy of these three drugs, right? Drug A has in this figure, drugs A and B have the same efficacy. You can see now, this is the outcome of it. Drug A and drug B have the same efficacy, right? But there is a difference in the potency, right? I think by this graph you will get the answer of the previous question what I asked you. So drug A has a greater potency than B, of course, because drug A requires less dose than the drug B, right? Suppose that drug A is 10 milligram, then drug B is 15 milligram, right? In the 10 and 15 milligram, they are giving the similar effect. Then which is more potent? Drug A is more potent than drug B, okay? While the efficacy of the both drugs are same. Uh, B or C must be larger to produce the same effect as A. So now in that case, B and C should be used less doses to give the maximum effect. Although drug C has lower efficacy than B, it is more potent than B at lower dose drug concentration. Drug C has lower efficacy. You can see the drug B has lower efficacy, right? If you see the dose required by the drug C is the same what by the drug A, but it has not achieved the same effect what the drug A and drug A has achieved, right? So in that way, if you see, drug C has lower efficacy than B, but it is more potent than B. Now you understand? Drug B and C, if you compare these two drugs, then B and C, if you compare, B has more efficacy than C, but C has more potency than B because of the low dose required. Okay. So like that, you should understand the drug potency and the efficacy. Like that, we compare in the bioassays of the different drugs. Here also the graph showing the potency and efficacy. You can understand if the potency, if you want to read the graph potency, then if graph is moving to this direction, that reads about the potency, and if graph is moving on this direction, you know the efficacy of the drug. Okay. Here you can see, here also three drugs have been shown, three different drugs, and showing the efficacy of that. If you see in the middle, EC50 has been shown. So here also the similar question comes. A blue, green and red, which is more potent? You answer me later, which is more potent and which is more efficacious, and compare between three, blue, green and red, and tell me the comparative efficacy and comparative potency. Here also shown by the another three combination, another three drugs. Here also you please tell me that the drug blue, green and red, which is more efficacious, which is more potency. Okay, here also I need your answer. Now if you see this one, another graph below, here its answers are given: equal efficacy, curve A and B; equal efficacy, curve C and D; equal potency, curves A and D. Now you see, equal potency, curve A and D. Why? Because of the similar dose or similar drug concentration. Curves B and C also have the similar potency, B and C, because of the same dose, both are required, right? So like that is a very wonderful explanation of the potency and the efficacy of the drug. Like that you can compare all the drugs. Later if you have any doubts regarding this, please first you understand this whole concept. If you have any doubts, please post me on. So activation of membrane receptors and target cell responses is proportional to the degree of receptor occupancy. This is the one concept. However, the drug concentration at which half of the receptor is occupied by ligand is often lower than the concentration required to elicit a half maximal biological response. So same thing. Sometimes the effect, the 50% effect of the drug you have obtained, but maybe you need less than the half of the receptors to act, to occupy it, right? So like that, this concept is also established here. If you want to see, you can see here that the 50%, you see the 50% response is obtained when the 50% receptors have been occupied. Okay, and here you can find a linear relationship. 50% receptor occupancy produces 50% responses, right? And but here is a non-linear effect has been observed. Less than 100 effect will be obtained by the less than 100 of receptors here, right? You can understand in this way. So I hope you understood that what is happening here. The 50% response receptors been occupied to give you the 50% effect, and the same 50% receptors are giving the 100% response as well. You can see this, you can see this here. 100% comes, right? But receptors have not occupied 100% to give the 100%, while only 50% has given you the 100%. So like that, you do not need to, that all the receptors should occupied to give you the maximum or optimum effect of a drug. Here one very important concept or the point has been mentioned. Because of this hyperbolic relationship between occupancy and response, maximal responses are elicited at less than maximum receptor occupancy. That I already told you. A certain number of receptors are spare. Spare receptors are receptors which exist in excess of these required to produce a full effect. Now you understand one point very important here. Receptor here, 50% receptors occupancy has given you 100% effect. Then, and there are 100 receptors available. So now what happened? You have some spared receptor with you. What are these? These are all the spared receptors, extra receptors you have, called spare receptors, right? It's a very important concept called the spare receptor concept. So a certain number of receptors are spare. Spare receptors are receptors which exist in excess of those required to produce a full effect. There is nothing different about spare receptors. They are not hidden or in any way different from other receptors. So they are similar thing, only they are extra than what you need. Here you can see one on the basis of this effect and the concentration, one formula has been derived, which is called the, where E is the effect observed at what concentration? At concentration C. So at concentration C, what effect has been observed, right? That formula gives you the, if you want to calculate the effect. E max is the maximum response, and EC50 is the concentration of drug that produces 50% of maximum effect. So E is equal to E max into C by C plus EC50. This is what the formula. Here one graph is there to tell you the effect of the agonist and intrinsic activity of the drugs. You can see the first graph, which shows you, this one shows the norepinephrine drug, right? Norepinephrine drug, and here this one is the phenylephrine drug. So these two drugs have been shown their response on this is a real graph has been shown. So what you are getting, you see the effect of this is a log dose versus response has been plotted. So now by using the previous concept, you can understand this very well, and then you can tell me which norepinephrine is more potent or phenylephrine is more potent, which is more efficacious, right? That is again a question on the previous concept, right? Once you understand that one, you can answer this. Here also the spare receptor concept has been explained well. You can see the drug A, drug B, drug C, drug D, drug E. There are 5 drugs have been used, and they all have been drawn a one EC50 value for them. So every drug has the EC50 has been shown. Okay, but if you see, they are different on that log scale. Their dose and the concentration of the drug, if you see in terms of concentration, the concentration has been changed. Drug A has very minimal concentration, B has little more, C, D and E have almost same. But if you see, the efficacy of all these drugs have been different. Means A, B, C have the same efficacy. Okay, so their efficacy is same, ABC, but their potency is different. A is highly potent, B is less potent, then C is less potent comparatively, right? But they all have the same potency. But compared to D and E, if you compare C, D, E, then only the what called efficacy is changed, potency is same, all are having the same potency. Okay, so these are very wonderful graphs to make you understand about these things. Now we'll talk about the you understood by the what the graded responses are and how to a drug and receptor means gives you the drug receptor interaction and the drug receptor interaction curves. How can you draw by the graded responses? Now I'll tell you what the quantal relationship. What do you mean by the quantal relationship here? So in the quantal relationship, what happens before here? What happens? You define a response that you want to check the drug's effect on this particular response, right? You want to see that if 50% receptor, 50% patients get treated, I want to compare the drugs on this basis, right? So this is what you have already defined the quantal or the quantum of the response. So when you already define predefined responses, if you are comparing, that is called as the quantal relationship, right? So we'll see what happening in the quantal relationship here. You can see they have taken the response of the phenobarbital drug. Phenobarbital drug is been used for the convulsions on the rat, right? Convulsions or fits, you can call it in common term, in the in among the rats. So what happened? You see that there are two responses have been shown, response A and B. And response A, where they have compared, they have already defined how much protection they want to check, that 50% protection of the animals, right? So 50% protection has been achieved by what dose? Maybe around 4 or 4.5. Here if you see, at the dose of 4.5 mg per kg, 50% of the rats get relieved of the convulsions, right? So this is already a predefined response you wanted to check. Second graph is giving you, in second graph, what you have decided? You have decided not this, not the what called cured rats, but you want to check on the dead rats. So what happened? You want to check that at what dose, what maximum dose 50% of the rats died, right? 50% of the rats died. So what dose it is? You can check it here, maybe somewhere 45, right? 45 mg per kg if you give, then you see LD50 is observed. LD50 means is what? Lethal dose 50, right? And here what was your observation? It was effectiveness of the drug. So ED50 was observed, right? Like that, you define the effect, quantum effect, and then you can check it. That's called the quantal responses. Now another important term to introduce with you, that is called therapeutic index, by which effective dose can be calculated. Thus, one estimate of a drug is margin of safety is the ratio of LD50 and ED50. This is called therapeutic index. So very simple. Previously, I have shown you what the ED50 is, what the LD50 is. So if you divide the LD50 by the ED50, you will get a therapeutic index, right? The therapeutic index for phenobarbital used as an anticonvulsant is approximately 10. By this, if you calculate, it will come approximately 10. So therapeutic index, a very important parameter to define the drug's efficacy, right, and the safety as well. Here also therapeutic index describes the relationship desired biological effect and toxicity. You can see the therapeutic ratio. If you want to calculate the therapeutic ratio or therapeutic index, then toxic dose 50 divided by effective dose 50, you will get the therapeutic ratio. Margin of safety, if you want to define, then lethal dose 50 divided by the effective dose 99. Effective dose 99 means what? You can see here effective dose 99 means at the concentration 99% patients 99 patients have been recovered, but one may die. So that's called the effective dose 99, right? So like that, these calculations can be done on the basis of the therapeutic index, right? Later I will explain you in detail about the what do you mean by the therapeutic index, what do you mean by the therapeutic level, subtherapeutic level, toxic level, right? These all things will be explained you later. So we'll summarize it here, how the graded responses can be. So more common than the quantal dose responses relationship is the situation in which a single animal gives graded responses to graded doses. That is, as the dose is increased, the response increase. Means by on a single animal, single patient, you can record these graded responses by giving the different doses. With graded responses, one can obtain a complete dose response curve in a single animal. Right, the data are typical of what one might obtain from constructing complete dose response curve in each of 5 different guinea pigs. For example, they have given a one graph by using 5 different guinea pigs and using this different doses. Next slide, I'll show you that graph. Since an entire dose response relationship is determined from one animal, the curve cannot tell us about the degree of biological variation inherent in a population of such animals. So this is the one problem with the graded responses, that you will definitely get a graded response of increasing doses on one animal, but inter-individual differences, right? Maybe the this amount of drug showing this amount of effect in one animal, maybe not in the other animal, because of the different body, because of the different biological conditions inside, different inherent system inside, right? So like that biological variation we are not going to get if you are using the graded responses. You can see here they are using 5 guinea pigs A, B, C, D, E, and using the different doses here, right? So you can see 0.001, then 0.01, then 0.1, then 1, then like that they have used. The drug here they have used the levoterenol. So what happens if you give the levoterenol 0.001 microgram per kilogram, then what happens? What the heart rate you will get by this, right? So heart rate increases by this way. You can see by giving all the doses 0.01, 0.11, you are getting the similar responses. That means similar heart rate, but the doses are increasing. It means the potency and efficacy, you can define that which dose is the highly potent dose and highly efficacious dose. That's what you can define by this. And by the previous concept, I think you just understand this and define it here, that which dose is the highest potency and which dose has the highest efficacy. You can compare between the doses, right? Now the last thing what I want to tell you, there is a one term called desensitization and tachyphylaxis. So what happens after some period of time or in some conditions, the receptors get desensitized. Receptors are not able to bind further, or not able to give you more responses, right? Even the drug is available is more. If the drug available is more, even though receptors are not giving more responses, that's called one level called as the saturation or called desensitization. So what is it mean? So all these terms, desensitization, tolerance, refractoriness, drug resistance, this all happens. How it happens? All these things: change in receptor, loss of receptor, exhaustion of mediators, increased metabolic degradation, physiological adaptation, active excretion of drugs from cell. Here I am not defining all this, just giving you this idea. Later in the presentations, next presentations, you will understand automatically that how the desensitization and tachyphylaxis achieved by these receptors. So I hope this concept what I have explained you here, you have understood well, and that's what I have gone little slow on this, so you can understand each and everything, because without these concepts it will be very difficult to understand further when I will start talking on the receptors and their mechanisms, right? So I have used these references for making this presentation. I think all these soft copy of the books are there with you, so please refer to them. Thank you very much for your time and listening it carefully and honestly. Please go through properly and post me on with the doubts. Thank you.